Hello everyone, my name is Caitlin Lang and today I will be presenting on polio. So what exactly is polio? Polio is formerly known as poliomyelitis and it belongs to the Picornaviridae family. This family of viruses is known for their icosahedral capsid symmetry as well as single-stranded RNA as their genome. They also are non-enveloped viruses. So polio is a virus and it is highly contagious. Before we go into the characteristics of the virus, I do want to talk about some of the history behind polio. There are plenty of contributors in research behind polio. However, there are four key contributors that have made the biggest progress. In 1904, Ivar Wickman is responsible for discovering the contagious nature of polio. In 1949, David Bodine discovered that there are three types or three strains of the poliovirus. In 1951, Jonas Salk, who is the most commonly associated with the poliovirus, created the OPV vaccine, which is a live weakened virus inside the vaccine. And in 1960, Albert Sabine was responsible for creating the IPV vaccine, which uses a kill vaccine. So, what are the symptoms of polio? Okay, so the symptoms vary depending on the person, depending on each case. There can be mild symptoms, no symptoms, moderate, or severe. Most cases, however, are asymptomatic though. And it's pretty rare. About less than 1% of cases actually experience paralysis, which is most commonly associated with polio, which is kind of a myth. Most cases of polio are asymptomatic. However, you can experience in moderate cases flu-like symptoms, which could be like a sore throat, nausea, stomach ache, tiredness, anything of that sort. In severe cases, however, like I said, paralysis can occur. In 1 out of 25 severe cases, meningitis is also found, and the feeling of paresthesia, which feels like pins and needles in your limbs. There are three types of paralytic poliomyelitis, and that includes spinal, bulbar, and bulbospinal. Out of the three, however, bulbar has the maximum potential for fatality because it destructs the cells of the brainstem. So how does one get infected? It is very contagious, so person-to-person contact is the first thing that comes to mind. However, the most common is through the fecal-oral route. So this could be direct contact with infected feces and somehow getting it into your mouth, or it could be infected feces on an inanimate object and you touch that object and somehow get your hand in your mouth. But basically the process begins with ingestion of the virus through your mouth, and then it multiplies and replicates as it moves through your gastrointestinal tract. If it does get worse, it does get into your bloodstream and can travel all the way up to your central nervous system, but that is the most severe cases. And this virus is restricted to humans, so no animals can be carriers or animals cannot be infected. So the viral characteristics of the poliovirus is that the genome is positive single-stranded RNA, and it also has a non-enveloped capsid that contains about 60 protomers. Each protomer contains 4 virion proteins, and it does have an icosahedral symmetry just like all of the other viruses of the Picornaviridae family. And in the picture you can see those little divots in between the plateaus. Those are called viral canyons. And those canyons is where the CD155 receptor of the host cell binds to the virus, and that's when attachment occurs. So how exactly does the virus affect the central nervous system, if it ever does? There are two methods of entry into the central nervous system, and could be through the blood-brain barrier, or it could be through the peripheral nerves. So when it comes to the blood-brain barrier, They don't know the mechanism quite yet, but they do know that it is independent of HPVR, which is synonymous to the CD155 receptor. They did a couple tests on mice to see whether increasing or decreasing the amount of the receptor increased or decreased the amount of viruses found in the blood-brain barrier, and they found that they had no correlation. So we know that it is independent of HPVR concentration, however, we still don't know how the virus actually gets inside. However, with the peripheral nerves, we do know that that process is dependent on the concentration of HPVR, and we do know the mechanism that it uses retrograde axonal transport, which means that the virus moves up the axon and towards the central nervous system. And the picture to the right shows the process of viral attachment all the way up until viral release. And you can see the single-stranded RNA being combined, and the single-stranded RNA is actually used as the mRNA for translation. And most often than not, the cell does lyse as a process of release. So the cell does die. So there are no treatment options for polio once you do have it, because there has no cure known for it yet. However, you can prevent from getting polio through vaccination, which I'll get to in just a second. But in the older days, when polio was just being introduced to the world, people were using heat packs to calm the muscle spasms, as well as frequent mobilization to just keep moving to prevent any kind of muscle deformity. Newer methods, however, have come with orthotic devices such as mechanical braces or anything of that sort, and they also have surgical methods such as tendon transplants or anything, joint replacements of the such. And a picture to the right is actually a picture of the iron lung machine, which was developed sometime during the mid-1900s. And it was for the patients that had respiratory failure due to respiratory paralysis. So it's an assisted respiratory machine, and it helped them breathe, obviously. And but it's no longer used because of the cost. The technical issues, is having a lot of technical issues, and it was also obviously just very large, so it's just very inconvenient. So talking about vaccination options, there are two types. There's OPV and IPV. OPV is a live and weakened virus. It is also more virulent than IPV, but I will also get into why that's not so much of a good thing either. IPV is a killed virus. It is inactivated, so the virus is not live. But OPV is actually no longer used in any polio-free region, including the United States, because of how many issues that have come up with vaccine-derived poliovirus. There has been an increase in cases, which I'll actually talk about in just a second, but OPV has been linked to an increase of vaccine-derived polioviruses throughout the world where OPV is still used. So OPV is actually trivirulent, monovirulent, and bivirulent, but IPV is only one type trivirulent. So the compass of OPV is a little bit more than IPV, but because it is a live virus, it does create a lot of issues and a lot more risk for increasing the vaccine-derived poliovirus. So right now in the world, polio has decreased about 99%. So in 1988, there was reported about 350,000 cases of polio, and in 2018, there was a report of about 30 cases of polio. So it has decreased quite a bit. In 1999, wild polio virus 2, type 2, was eradicated. In 2012, however, complete polio eradication was declared a programmatic emergency by the World Health Organization because of the steady increase of the vaccine-derived poliovirus. So they want to stop using OPV as much as possible so that we can move away from the vaccine-derived polioviruses and take care of that. But by 2013, only Nigeria, Pakistan, and Afghanistan are considered polio endemic. So I wanted to talk about WPV versus CVDPV, wild poliovirus versus circulating vaccine-derived poliovirus. Obviously, one is a natural strain and one is derived from the vaccine. And some of my sources.